Hello everyone, welcome to Chromosome 21, and today we'll be looking about genetic mapping of genes, distribution of genes on chromosomes. So let's get started. So before we move, we would look into some basic terminologies that will be used very frequently in the presentation. So these are very basic terms. You can go to Google and get information regarding that, but we'll be discussing it here anyways. So meiosis is one of the two types of cell division. Second is mitosis. Meiosis is exclusive to sexually reproducing organisms, where the genetic material is reduced to half, and they form gametes. After formation of gametes, two gametes fuse together to form the new individual, where the genetic composition is reconstructed. And recombination and chiasma are two uh, occurrences that happen in meiosis. These phenomena leads to the physical exchange of DNA material between two sister chromatids of homologous chromosomes, and this leads to better diversity and recombination of genes. And this will be looking into because this is very important from the perspective of genetic mapping, and chiasma is the actual physical phenomena that leads to recombination. This is the phenomena where there is a intersection of two chromatids, and there is an exchange of genetic material. We would also expect that some basic pedigree analysis, what is a pedigree chart and how to read it. So because we'll also be having that in subsequent slides, so pedigree chart analysis is somewhat handy in this occasion. Locus is genetic locus, by which one will mean that there is a specific coordinate or a specific location on the chromosome where you want to look into. It is a very specific thing, and you don't want to highlight the whole region, just a specific nucleotide. So this is locus, and when you want to specify a region, you want to call it as a region, not a locus. Linkage or linked genes are two genes, those are present on the same chromosome, so they are inherited simultaneously from the parent to progeny. And genotype and phenotype are the two characteristics or two states of an individual. Genotype refers to the genetic composition of the individual, while the phenotype refers to the observational or molecular characteristics of the individual. So, having these terminologies in our hand, let's move to understand why do we even need genetic mapping. Genetic mapping is, in one word, we can say a map where we know which gene lies where. So, like a physical map, what we have in our hand while we navigate throughout the world, we know where to take the turn, and we know where each of the component of the map or the surrounding lies within. So, we have a physical visual map which you can reference to when we are lost. But when we could talk about genetic map, we are talking about something very similar. We are talking about where the genes are placed on a physical linear dimension. So we have this whole chromosome, let's say, and we want to know where our genes lie or what are the genes that are present on the chromosome. So this is why we would do genetic mapping. But let's say, why do we even need this map? Right, like why is this important? Why do we need this map ultimately? Even we need this kind of maps because of various of things. Like historically, when it was first determined, it was of course like enigma that where the genes lie within the chromosomes, and then later on it was developed that we can also manipulate our genetic composition with the help of various tools and techniques. And there we better understood that there is an increasing need of genetic maps. But historically, it was done to identify the causative genes, disease causing genes that are present in our chromosomes, and exactly where they are located. We ultimately create some database like OMIM, where you have all the information regarding some your genetic diseases and disorders, and have a very handy and compact information catalog. And we also create something, this is called a genome browser. This is UCS genome browser, where you have a physical map of everything, of every gene on a chromosome, of every exomes on a gene. So you have this beautiful linear map, and you can reference it to when you want to look into the coordinates of the gene. And you would also want to look that which genes are subsequent to other ones, so that you can create some genetic manipulations. So those are very higher concepts, so you can look into it. And we also want to look into the inheritance patterns, or the inheritance of our disease or gene of interest from within a family, from parents to progeny, and ultimately understand, if you don't know, ultimately find out what is the causative element of that disease. So genetic map comes very handy in genetic disorders. And talking about genetic disorders, classify them as polygenic and monogenic disorders. So polygenic disorders are the kind of disorders which involve multiple genes for disease to occur. And examples of these are several, like metabolic disorders like diabetes, we have neuropathies, hematopathies, etc. And these are more complex than monogenic, which is the other type of genetic disorder, as they have several elements to confer a genetic disorder. We would need multiple rounds of evaluation, and also it is a bit more complex. While on the other side, monogenic disorders are much more easy to understand. We know that there is a specific gene that causes a genetic disorder, so it becomes much more easy to get hold of that gene. So examples include Marfan syndrome, Huntington disease, which is caused by Huntington gene, Marfan syndrome caused by fibrillin 1 gene, neurofibromatosis, and sickle cell anemia, etc. There are various monogenic disorders. Genetic recombination is very helpful in such kind of genetic mapping procedures, and this is the basic crux of, it's like underlying whole environment which leads to the genetic map formation. So let's understand about genetic recombination and linkage before we move to the subsequent slides. Genetic recombination is nothing but the physical exchange between two sister chromatids of two homologous chromosomes. Again, sister chromatids and homologous chromosomes, I hope it is very clear. But for now, we know that there is something called bivalent. So this is one homologous chromosome. This is another homologous chromosome. These are the same chromosome. Let's say chromosome 21. So this is chromosome 21 from paternal side. This is chromosome 21 from maternal side. And these two are sister chromatid for the same homologous chromosome. This is the paternal side, two sister chromatids, maternal side, two sister chromatids, and then there is a chiasma formation between these two chromatids, while these two chromatids remain intact. Ultimately, we get a non-recombinant, a recombinant, a recombinant, and a non-recombinant. Two of the recombinants and two of the non-recombinants. So as I mentioned, genetic recombination is very important in understanding the genetic map or genetic mapping. So how is it important is that we have to understand that this genetic map is actually the probability of recombination between two loci of our interest, and one of it is that we already know it exists in the on the chromosome and on a very specific place on the chromosome. So no, we know its location, it hasn't been predetermined. But the second one, we don't know. We are mapping that exact disease on the chromosome. So we will be creating a map between these 2 points, like in a physical map where you have a reference point, and then from there you can move onto the map to some later places where you want to go. For example, if you have a tree or a house, and you can exactly pinpoint that we have to go left and 20 meters from the house or from the tree, exactly like that, we have a reference point on the chromosome from where we want to map that house or the next location to, or our disease gene or our gene of interest in this case. So genetic recombination is exactly this, finding this distance. And to find this distance, we have to get the probability of recombination between these two physical locations. So to give again a very vague analogy, is that if there is a random line drawn on the map, and what is the probability of the line going through that path that connects these 2 points, the house and the location, that will be determined by the actual distance between these two objects, right? So if they are very close together, there will be very low probability of a random line crossing them between them, while if they are very far apart, there is a higher chance that there will be a random line that will cross between these two points. So if, exactly like that, if there is a genetic recombination occurring in the in the chromosome, and if the 2 points of interest are very far away, then there is a very high chance of recombination. If they are very adjacent to each other, there is a very low frequency of recombination. And by observing this frequency of recombination, we can ultimately map where these 2 points lie, or at least relative to each other. So with that, we can move forward and understand that where these 2 points are actually located on the chromosome. Now, for an example, to take an example, we see that there are three loci on this chromosome: B, P, R, and VG. They are separated by some genetic distances. This will have to identify, this will have to calculate. We want to get to know these values, but what we know that there is a recombination frequency between B and PR, and recombination frequency between PR and VG, and we know that there is a higher recombination frequency between PR and VG compared to B and PR. So we already know that there should be a less genetic distance between B and PR compared to PR and VG, but this exact genetic distance is calculated from the recombination frequency. And how exactly, that will be looking further into the slides. So in our chromosome of interest, we have two loci, A and B, where there are two alleles for each of them. For A, we have A1 and A2. For B, we have B1 and B2. A1, let's say, comes from paternal side and A2 comes from maternal. B1 comes from paternal side and B2 comes from maternal. Now, again, these two are homologous chromosomes, and they have formed their sister chromatids. One of the sister chromatin with the other forms a chiasma, and there is a genetic recombination, and ultimately forms four different kind of gametes. So one have non-recombinant from paternal side, one have non-recombinant from the maternal side, and these two have two different type of recombinants. So these will give rise to four different gametes, which will fuse with the other gamete from the other partner and then form the new individual. So the new progeny may have non-recombinant type of chromosome or recombinant type of chromosome based on whichever gamete fuses. So these two alleles are crucial for our study, and there is a specific name to given each of these each of these loci. So this is a known loci, where we know that this loci is present at the exact this point of chromosome. So we know the coordinates, while this query loci is something that we don't know of. So we will be mapping this loci relative to our known loci. And here it is important to mention that when we talk about recombinant and non-recombinant, it is always in perspective of our these two loci. It's not that if there is a, let's assume that there is another locus present here, but for this for this recombinant that we are saying, if with respect to this locus and this locus, it is a non-recombinant because there is no recombination occurring between these two. So for a gamete to call as a recombinant, we have to have a recombination event between these two, or the query and the known locus of our interest. So after that, we can call it as a recombinant or a non-recombinant. But if there is no recombination between the loci, then we cannot call it a recombinant. So this locus, where we know its coordinate or its location, we call it as a genetic marker. So genetic marker is a set of known genetic loci that is used as a relative position for determining the next genetic or the gene of interest in our chromosome. So let's understand the genetic markers in a bit detail. Genetic mapping with markers is one of the most crucial steps in genetic mapping, and markers therefore hold a very crucial role in this phenomena. Genetic markers are, as I said, unknown locations on the genome, and by far there are several genetic markers. There are several types of genetic marker, family of genetic markers. These are very few example of genetic markers that were characterized in chromosome 12. So these are the markers that we know that are present in our chromosome 12, and then related to these we can find our gene of interest location on our chromosome 12. Now again, these genetic markers can only be used to map genes that are present in the chromosome 12 and not on any other chromosome, because then those genes are not linked. So there it comes the linkage problem, that we can only map the linked genes. We can only find the genes that are present on the same chromosome. So you cannot, like, there are two islands and there are two different maps altogether. So you cannot just point at one map and say that there is something, there is another location on the second island with respect to this. Now, one may argue that we can define a locus on our chromosome by ourselves, and then we can find the relative distance of a gene of interest compared to that loci, and then we do not need these genetic markers. So why don't we do that? Well, there are a number of features that are needed for understanding or for getting that information from a specific point of chromosome to your next gene of interest, and that locus should have a very clear Mendelian inheritance. So you should be able to determine the presence or absence or the state of that locus based on some kind of phenotype or some kind of measurable characteristics of that individual. And you can not just measure, but you can observe the inheritance of that character throughout, and therefore you can conclude that what is the state of the genetic locus. And that's why it is very important that locus shows that there is a clear Mendelian inheritance, and preferably dominant, because then of course you would not need to have a homozygous recessive individual just to observe the phenotype. But for some markers it is impossible to have that kind of phenotype, so it is preferably dominant. And it should be well characterized. Of course, you should know its location, you should know its exact sequence, and where in the chromosome it belongs to, and what are the adjacent genes it has, so that you can know where exactly you are looking into the chromosome. And the third and probably one of the most important characteristics of this locus should be that they should be highly polymorphic in the population. What is polymorphism is, for a single locus on a chromosome there should be polymorphism of that locus. So in one individual they might have different sequence for that location, on the other individual it might have different sequence. So the sequence may differ, but the relative position of that marker will stay same in respect to the chromosomal length. So this polymorphism is very, very important when you look into different kind of meiosis that occurs in the population. Now, one may say that how do we know the location of the markers in the first place? Like, we are creating a genetic map to understand the location of the some of our locus, but then you are saying that we need a predefined or a pre-known, already known locus related to which we will be finding the distance. But how do we get to know that exact first location in the first place? So historically, we didn't even use these genetic markers. We use some other kind of markers like HLA tissue types or blood groups, and that is how we linked our disease to a specific place in our chromosome. Historically, we did not start with a single point mapping. So when I say that there is a set of loci, known locus and a query locus, we have only one query locus, so it is a single point, right? But in historic perspective, we did not start with a single point mapping. We started with a 2-point mapping, and this will not be 2-point mapping will not be covered in this video, but we'll be having a separate video on that. And 2-point mapping was the first way, because you will be having two query locus, because we don't know the location of either of those. Therefore, you will be finding the distance between these two locations. That is how the historically it was done. So, and that is why when you have two set of unknown loci, becomes very difficult, and therefore this set of genetic markers was established, because now you already know the location of one set, locus. So just related to that, you can find the second locus. So moving on, let's take a quick example about the events that I was talking, so that we can have a better understanding. So if you look into this example pedigree, you can see that generation 1 individuals are deceased. So we cannot find the genetic constituents or the genotype of these individuals. But for generation 2 we can. And out of that, the father is diseased, while some of the progenies are also diseased, and rest of them are healthy. And we see that there are two loci in the chromosome that we are studying. One is A and one is B, and there are two types of allele present for each of the locus. So A has A1 and A2, as you can see here, B1 and B2 for the B, and father is heterozygous while mother is homozygous. Now, for father, we know it is disease, he's diseased. So we don't know if A1 or A2 is causing the disease, B1 or B2 is causing the disease. So we'll have to find out which one of them caused the disease. And ultimately, when we look into the pedigree, A1 is also present in the mother, and that too in homozygous form, but the mother is not having any disease phenotype. So we can elude out that A1 is not involved, or B1 is not involved in the disease outcome. Therefore, A2 and B2 becomes the obvious candidates. And looking into the progeny, we see that the individuals having A2 are having the disease phenotype, while A1 do not lead to any disease phenotype. Now, this is only possible when we encounter for recombination. Why so? Let's imagine that there is no recombination. So we'll have only these kind of individuals, where A2, B2 and A1, B1 is present, where A1, A1 and B1, B1 are present. We have again these kind of individuals. We have again this kind of individuals, and A2, A1 and B2, B1, these kind of individuals. So we have only these two kind of individuals when there is no recombination. And this is exactly what the father has. So we don't know, still we don't know that A2 or B2 cause the disease. But when there is a recombination, this time these two have exchanged with each other, and we have A1 and B2 with A1 and B1 from the mother. Now, this time, as we know that A2 is not present in this individual, which is from the paternal side, this individual does not show any phenotype. Similarly, where there is A2 retained and B2 is exchanged with B1, you see that there is a disease phenotype. So we can say that A2 is associated with disease and B2 is not. This is how we have associated a disease to a specific marker or a specific location on a chromosome. And it is possible only by looking into the recombinants. And this mapping is a subtype of genetic mapping, where you find out the causative gene or the disease gene that leads to the disease phenotype. And this has its own protocols. We'll be looking into some other videos, but for now, let's look into informative, uninformative, where this pedigree was very informative. Not all the pedigrees are as much as informative that this pedigree gave us. So there is something called as Polymorphism Information Content, or PIC, which is obtained from a specific pedigree, and this helps us to understand, or this helps us to map our loci on the chromosome. Now let's take this example. So where this individual has a homozygous genotype, which is passed on to its progeny, and it is also homozygous. So from this we can elude out that this person were contributing A1 allele, because one A1 came from the maternal side and one A1 came from the paternal side. And this does not say us which of the A1 came from the maternal side. So this does not give us the information that which gene was inherited from which parent. So this is not very informative to us. Similarly, when you can see from here, from this generation to the subsequent generation, this time we see that there is a A2 and A1 and A2 allele, and this time we can correctly identify that A1 was coming from this individual while A2 was coming from this individual. And this time this is informative, but the whole pedigree is not very informative, because we don't know A1 from came from the grandmother or the grandfather, paternal grandmother or paternal grandfather, we don't know. Come to second pedigree, we can say this time that okay, this individual contributed A1, this individual contributed A2, but the individual that this father married to has the same genetic composition. So this individual received this A1 from her mother or her or from her father or her mother, we don't know. So again, this is not very informative. Come to the third pedigree, we see that there is a again similar kind of issue as we occurred in our first pedigree, that we don't know from where this A1 is coming from. And in the second generation we have the problem that this pedigree faced, which is we don't know where this A1 came from. So this A1 can come from her mother, her father, her paternal grandmother, or even from her paternal grandfather, we don't know. So this is one of the most uninformative kind of pedigree that we can have. But if these loci did not only have one but several kind of alleles, that is, if they were highly polymorphic, what would happen is, even if the paternal grandmother was homozygous, we know which gene came from where. So this father received his A1 share from his mother, A2 from his father, and because he married to a person who had different genetic composition than his for the same locus, by the way, this is for the same locus, he has A1 and A2 while the mother has A3 and A4, we can say that progeny received A2 from father and A4 from mother. So if we want to track A2, we can say this A2 was received from a father who received it from his father. So we can track this A2 all the way to his paternal grandfather, while this A4 can be mapped to her mother, while then we can look into their own pedigree from his maternal side, and then we can find out where this allele came from. So this is the most informative kind of pedigree that is in our hand, and this was only possible because we have polymorphic variant of this locus. And this is exactly what I was talking about when I said that genetic marker should be highly polymorphic, because if it was not, we could not track our alleles to our ancestors, and therefore we cannot find out if it was recombinant or it was a non-recombinant. So now we know that recombination is very important for getting the genetic mapping done. So we have to now understand how it is done. As I said that genetic recombination translates into the genetic map, but this is exactly not equal. Looking into one such example is that when you have several loci, all of them 5 centimeters apart, end to end distance becomes 75 centimorgan. But when we translate centimorgan to recombination frequency, it roughly translates as 1 centimorgan to 1% of recombination frequency. So 1% of recombination in the whole population. So when we see that there is a 75 centimorgan distance between two loci, you cannot say that there will be a genetic recombination of 75% between these two loci, because linked genes cannot have recombination frequency of more than 50%. And therefore this does not add up. And this was exactly what was argued by Castle, that genetic mapping was non-linear and it cannot be mapped using mathematical functions, and that there has to be some other kind of effort to be made for getting a genetic map, which was against what T.H. Morgan was proposing, what suggested that there is a linear relationship between the distance, physical distance, and recombination frequency. And this was then later solved by JBS Haldane by giving a mapping function. What is a mapping function? Mapping function is a mathematical relationship between recombination fraction and genetic map distance. This created a solution. This gave a solution to the ongoing rivalry between Castle and Th Morgan, and this gave a direct relationship between your recombination frequency and your genetic distance. So what Haldane found, and JBS Haldane is the person you might have heard about in the population genetics relations, but JBS Haldane was originally a mathematician who first dived into the genetic mapping business, if you say, and then later he was into the population genetics. So originally he proposed, what he observed was that up to 10% genetic recombination you can roughly translate into the genetic map into linear relation, like 1 centimorgan roughly translates to 1% of recombination frequency. But when you move beyond 10%, it reduces. So 11 centimorgan roughly doesn't it doesn't exactly translate into 11% of recombination frequency, but somewhat less than that. And again, when you do it into a graphical plot, you find a logarithmic relation. So you can see that 45 centimorgan will be translated into 30% of recombination frequency and not exactly to 45%. But when you look into the realistic values of genetic maps, it doesn't exactly correlate with even this function. And later on, it was found that this is because something called interference. Interference is a phenomena where there is a inhibition of another chiasma formation with very distance, with very close, within the very close proximity of already formed chiasma. So, and as you increase the distance between 2 points, a chiasma formation probability also increases. So, due to this inhibition, this adjacent chiasma formation inhibition, double crossover is very improbable, very improbable, and that is why we have to take account of that as well. And later on, the Haldane's genetic function was again defined by someone called D. Kosambi, and he proposed that this new function would better correlate genetic recombination with genetic distance. And this becomes a bit more complex, so we will be not diving into the mathematical derivations and other aspects, but Kosambi's mapping function takes account of interference and gives a better recombination, it gives a better genetic map distance compared to what Haldane's equation gave us. So what does all of this mean? What do we get out of all of this? What is the outcome? What is the output of all of this? This mapping function, this genetic distances, recombination frequencies, etc., etc. We get genetic map. Genetic map is as explained, it is a physical form. It's a tangible form of our chromosome, the the gene distributions, and to some extent understanding where and how much recombination will occur in that chromosome. After doing all of that hard work, we get two kind of genetic maps. One is physical map, known as genetic map. Another one is linkage map. These two are linkage map for two different sexes for a single chromosome, that is chromosome 13. And it shows something very interesting. When you look into the physical map or genetic map, you see that there is a uniform distribution of nucleotides. Like these units, one unit, each unit resembles this single unit resembles a unit distance from each other, and it is uniform. So let's say there is a distance of 500 kilo base pairs, and this is another 500 kilo base pairs, these two. Now one would argue that the recombination frequency would give us let's say 5% recombination frequency here, 5% recombination frequency here, and even though not additive, we would get somewhat 10 or let's say 8 recombination frequency. So okay, let's fine, it's okay, we got 8 centimorgan, okay. But that's usually not the case. When you see these two units, let's say again 500 kilobase pair, 500 kilobase pairs in genetic map, but when you see the recombination frequency, there is no recombination in this whole part. It is almost identical in parent and progeny and subsequent generations. So there is not much of recombination happening over here, but in this tiny little space, so this whole thing just goes in one block, and then the just next block is very tiny compared to this whole block. So what this says is that even though genetic map or physical map is uniformly distributed, the linkage map is not. And this leads to formation of very tiny or very large blocks that is passed on from generation to generation. And there is a varied recombination frequency within these blocks. So within the block, recombination frequency drops very low, while between the blocks, there is a very high recombination frequency, and these blocks are called haplotype blocks. Haplotype blocks are these kind of blocks in the chromosome where of course there is very low recombination, and within these two haplotype blocks there will be very high recombination. Now looking into this, we realize that recombination is not very uniform, and recombination is not a very good measure to get the genetic map. But then again, we don't have any other way as well. So we have to go with recombination and then later elucidate what is the physical map on that chromosome. Now it is not entirely useless. It will not get, you will not get the exact genetic distribution, but you will get the recombination distribution. So you get this recombination frequency graph where you know that if you have a marker here and you have a marker somewhere here, you cannot just go about linking these two in a study, and you cannot find a very good genetic map if you consider this marker and hair marker. So you, if your gene of query lies within this block, you should take a marker from this block which is very tightly linked to that gene query gene. So this gives a information that how much recombination is happening within that specific region of the block of that chromosome. So this gives another kind of map. Now this is helpful like physical map, where one physical map shows the geography of a terrain while other physical map shows the population of a particular city. And even though those two are of the same place, they give totally different information, and none of them are useless. They are useful in their own matters. So these two are useful in their own matters. And you see that there is a very high recombination frequency between these two blocks, and between these two blocks, but very low frequency of recombination within this haplotype block. So intra haplotype block recombination frequency is almost null compared to inter haplotype recombination frequency. So this brings us to the end of our presentation, and to conclude, what we have learned is that genetic maps are very crucial when understanding the linkage and linked genes. So we want a spatial distribution of our genes on the chromosomes, and for that we have to create a genetic map. For creating a genetic map, you need to understand the recombination frequency, and only by understanding the recombination frequency you get a recombination map, a linkage map, a physical map, genetic map, different kinds of map which you can then use for different purposes. Genetic mapping is necessary for disease mapping, so understanding which disease is caused by which gene, and of course it is different for different polymorphic and monomorphic genes, and therefore different segments can be associated with certain genes. Genetic mapping involves looking into genetic recombination between marker and gene of interest. As I have been saying it repeatedly, that it is always understood by studying the genetic recombination between marker and a gene of interest. And genetic distances and recombination frequency, all the equivalent, are not additive and definitely not equal. So this is again very well understood by now. And mapping functions, Haldane's function and Kosambi's function, are very good and still used function that gives us the mathematical relationship between your linkage map and your genetic map or physical map. And you can gain your own insights from those using those functions. So that has been all for this presentation. I hope you find it insightful. Thank you.